Extra credit. Let's go. So, you can read this. Read read this stuff. Let me move this over so I'm not hiding in the dark. There we go. All right. Stuff at the top, that's just like what we're doing and why. What you're going to be doing is finding a brand new virus. We already know where it is. We know a database. We know what kind of animal it is. And so what you'll have in the instructions is going to be an example that you can practice with. Do not build out the entire virus because I already have this virus, yeah. And it doesn't count as any extra points. This is just so you can know you're doing it right because that's going to be most, if not all, of your questions. Am I doing this right? Do the example. And if it works, yeah, you're doing it right. Congrats. Yeah. Okay, what I'm going to give you is going to look just like this, but it's going to have different numbers and different words. Yeah. There's going to be a database. So it's going to take the form of SRR, ERR, or DRR, and a bunch of numbers. Doesn't matter. Don't worry about it. It depends where the database was first housed. Yeah, it's not your problem, and it'll work anyway. Then it'll have the name of an organism. This is the organism in which the virus has been found. So these are people that were reading out RNA from the organism, and the organism happened to be sick, and they probably did not know it. Almost certainly did not know it. But we can find it, and we can build it. And it helps us advance our knowledge of nidoviruses, which are the group that coronaviruses belong to. So kind of a big deal, yeah. Over here, the third thing you're going to get is going to be the name of a RDRP seed. So this is the polymerase core that is going to be closest to the polymerase core of unknown virus from Ambystoma mexicanum. Google Ambystoma mexicanum if you want. It's an axolotl. Axolotls are amazing. Yeah, but it doesn't matter. You can do this without knowing what an axolotl is. Yeah. Every virus you build is going to get 50 points if it is complete, and it will be prorated. So if you give me half a virus, I'll give you 25 points. That's extra. That doesn't increase the total. So like if you're on 500 out of 1,000 points, one virus will get you to 550 out of 1,000. The next virus will get you to 600 out of 1,000. You can keep doing viruses, but you got other stuff in your life, and that's okay. If you can get even one of these things done, 50 points is half a letter grade in this course. That's a big deal. And I know some of you need it. Yeah, I just saw your test answers. All right, and even if you don't need it, whatever. Yeah, you're participating in research. You're doing a good thing. The virus that you find, once we get it cleaned up and do a bunch of bioinformatics on it, this may end up being the closest relative to the next big pandemic thing, and you will have helped. You don't know, because I don't know, and nobody knows, but yeah, why not? Why not try to help? Doesn't that feel better than not helping? I think it does. Okay, you're going to get the other number. It's annoying that we have to get the other number. It's in step 1. The way our program spits it out is this number. This number is fine, this SRR number or DRR or ERR. But half the time it won't work on all the other steps in BLAST. And so we've got to go get the magic master number, the experiment number. So it's going to be S, D, or E, R, X, and a bunch of numbers. And they're not going to be the same numbers because of course not. That's the number you're going to need. That's the number you're going to use, the number with the X in it. Yeah, so there you go. You're going to go get that. It's got the website you go to. You plug it in the box. You can do that. I believe in you. Here, if you want to see it, it's this website. So let's see, go back one tab. You go to this website. You put in the SRR number or whatever, and you get this. And there we go. That first thing right there, that's the SRX number. That's the thing you want to copy. And we're going to save that. We're going to save it in a document. The document is where you're going to build your virus. It's got instructions for how to make a proper little title for your virus. You're going to make a little FASTA title for your virus, and it's going to look great. It's going to take that form. And so for our virus, it's going to look like this. Boom. with all that information in there. And it's just a nice way of keeping all the information together so you don't build a virus and then say, huh, I wish I remembered where that came from. Because I can't do anything with it if I can't tell what it is and where it's from. So yeah, keep your records straight. All right. Let's see. You're going to download from Canvas this file. It is, well, it's not here because that's where I'm putting the video guide. It is right there, Nito RDRP seeds. That's where you're going to find the correct seed. There are like 500 seeds in there. Just, you need your one, none of the others. The names are close. Don't sue me. The names are not quite exact. And the sequences are proprietary. Some of these are not published, so don't go spreading this around, all right? Yeah, all right. So, next up in our instructions, you're going to use the nearest virus. You'll use the amino acids from the nearest virus to find matching sequence in your sequence database. And that is the sequence that we are then going to extend and extend until you have a full virus. To do that, you get this file. So I said this one is closest to Synnathus typhal coronavirus 2. You're going to do a find, look through the list. This one ends up being the correct one. Most of the time I've replaced spaces with underscores. Sometimes I abbreviate coronavirus as COV, but the elements will be there. Sometimes I'll leave off the species because I'm just going fast and I've dropped something. It's okay. I know where they come from. I can figure it out. I've got another bigger database of those. They're just not as clean as I would like. Yeah, and here it is. The colors don't matter to you. If you care, that STD is the active site, STD, and it's either that or GDD in this group of viruses. The yellow part is going to be the actual polymerase core domain that we're trying to match, and we're going to go fishing. We're going to see what we can catch that's going to be similar to this, but from your data set. And then that's the most conserved part. That's the part we're going to have the best chance of grabbing. And then from there, once we get the nucleotides that spell the protein that is similar to this protein, then we can start to build it out just using nucleotide searches and finding the piece that matches the end and then adding it on, finding the piece that matches the new end, and getting wider, wider, wider until you get the full genome. Full genome is going to be somewhere between 15,000 and maybe 50 or 60,000, but I doubt you hit one that big. I don't think any of these are going to go much above like 35,000, be my guess, but you never know before you build it. So I can't tell you how big it's going to be. I can't tell you anything except if it doesn't have a polymerase, RDRP, it is not a nidovirus. And yeah, all of these things have been pre-screened, so we know there is a nidovirus in the data set. We just have not built it yet, and you are doing this. The process is called Mechanical Turk. Yeah, look it up if you want. It's a thing. It's where you break up a task into little bits and have lots of people doing parts of it, and then like human power can actually beat computer power, and we do not have an efficient way to do this with computer power. The alternative is me doing all of these myself, and I'm old. It hurts my eyes. Yeah, so whatever. Okay. So we're going to go to tBlastN, and it's got to be tBlastN, not any of the other blasts. Just go to that link. Once you're there, you're going to paste this thing into the box, like so, and you're going to make some changes. Whoa, you're going to make some changes. So when you come in, it's going to say core nucleotide. You're going to change that to sequence read archive SRA. That's important. That's going to clear out this box, and you're going to say, oh no, what's the number I put in there? And the number is your SRX number, so let's just find it over here. Where did we put it? There we go. Copy that and pop that in there, and it's got to be the SRX number. If it gives you an autocomplete, you can click the autocomplete, and that's fine. Or you can just put in the SRX number, and it will work. Then we got to change some things. We got to click this so we show results in a new window. Otherwise, it sometimes breaks. You don't have to mess with this for right now, but this will be on when you come in, and you want to turn it off. Once you've done those things, you're ready, and you can blast. And so, boom, hit blast, but I've already done that. And so, when the blast comes in, if you get results, and you will get results because you're doing the same one first time that I'm doing, and so these are the results that you should see, or something real close. Yeah. All right, so I told it to cut off at 100. That's fine. These are the numbers. It says the length of each one is 150, and that's correct. This is the accession number of each one, and it's going to end in a 0.1 or a 0.2, and you're going to have to remember that. Yeah. From this screen, we can't tell which of these are forward reads and which of these are reverse reads. It describes both of these things with a little picture of a fish down here. There we go. So there's always a pair of reads, and they're hooked up like this. So there's a forward read on this side, and there's a reverse complement read, like a tail read, reverse read, on the other side. And so if you find the reverse read, you know there exists a forward read that's over here, before it in the sequence. And if you find a forward read, you know there exists in the other half of the pair a reverse read that is somewhere after that forward read. We're going to use that to be able to go fast. If you can't get your head around that, you can do this in little baby steps with only forward reads, but it will be absolutely painful and it will take 10 times as long. Yeah. With this, I can get through a brand new virus in about 2 hours on a Saturday morning. If you're trying it during the height of day, during the week, the service sometimes slows down. And if there's a government shutdown, that sometimes slows the service down a little bit too, although it's been pretty good for me lately, so whatever. If it's not working for you, try again on a different Wi-Fi connection. Try at home. Try at a different time. So yeah, 2 reads like so, called 0.1 and 0.2. The 0.1 is not always the forward read, and the 0.2 is not always the tail. It's like 50-50. You cannot tell from the name which end you've got, but you can tell from the results. So to tell that, we're going to go to the Alignments tab. So it's like halfway down the page right there. On the Alignments tab, we're going to see them all at the same time. So let's go to the bottom choice, flat query anchored with letters for identities, like so. And there we go. These are all of the potential amino acids. And it's going to show you all the first ones, and then you scroll down, you can see the end of it. That's fine. Doesn't matter which one we pick. For this one, I picked this read. Rather, what matters is you need to pick a forward read. Otherwise, your life will be difficult from the start. Don't make it difficult from the start. It gets difficult later. Yeah. A forward read is going to have a low number over here and a high number over here, like it's going forward. A reverse read is going to have a low number over here and a high number over here. So, yeah, you get it? Here. We can try it with any one. We can try it with this one. So, and now before I click this, I'm going to look, and I'm going to remember, like, oh, this read, this forward read ends in 0.1. So if I want the forward read, and I do, I need to click the 0.1, because it's going to show me both ends of the pair. It's going to show me just like this. Yeah, now do you remember which one was our forward read? Yeah, I do. Of course you do. It's 0.1. It's this. You can just double click in here. It'll select that. You do not need the title. Just you're going to copy this and you're going to bring it down and you're going to paste it in your document. And so there's the one that I pasted before. And so that is the seed, the nucleotide seed. Now you're done with that step. Nucleotide seed done. And so now the last step of the process, which you are going to repeat over and over again, you can get to the end in maybe 25, 30 searches if you are clever with how you search. And if being clever is too much brain power right now, you can get to the end in 100 searches or less, probably. Yeah. But you got to be searching both ends. If you're only searching one end at a time, it'll be like 200 searches. And that's okay. Yeah. If that's your thing. All right. So now you have your seed. Now you're going to do the next step. And for this, we're going to go to a nucleotide blast. So nucleotide blast is linked in the thing. There you go. Nucleotide blast. That'll take you there. And you're going to do similar to what you did last time. You're going to paste in your nucleotide seed. How can you tell this is nucleotides? Because it's got A, C, G, and T only. Whereas the other thing, that was protein, had all these other letters that are not only A, C, G, and T. There you go. It's pretty easy to tell. Alright, so you're going to paste your nucleotide sequence in this box. You're going to change whatever, the standard database to SRA. You're going to paste in your SRX number. You can click autocomplete if you want. You're going to click show results in a new window again. You can click either 250 or 500. I did it with 250, but you may, whatever. Either one will work. Bigger searches, like for more reads, take longer to run and longer to format. So you'll get a sense with your data set for how deep this virus is. And there may only be 25 reads that will extend what you've got. Yeah, in which case it doesn't really matter. You can put 100 in there and you'll still get all of them. But if you have really high sequence coverage, like 10,000 sequences for each little part, then you may need to go more like 1,000. Yeah. You will just have to figure it out, and it's not going to break if you pick the wrong number there. You'll still be able to inch along, but it's going to be going faster if you've got lots of sequences to pick from. That's what you want. Anyway, you're going to change that to a bigger number than 100. You're going to turn off these two things because they're silly. You're going to hit blast, and you're going to get a result that looks something like this. There we go. You're not going to be on descriptions page. You're going to click your alignments page. You're going to click the next to last choice, flat query anchored with dots for identities. The dots are going to show you whenever a read has like a mutation in it that's probably a sequencing error like this. If you get whole columns of mutations, then maybe the one that you had a sequencing error, and these are actually all showing the correct thing. Yeah. And that's fine. You can change it if you want. If you can't figure out how to change it, don't. Yeah. Avoid the ones with like just a single nucleotide that's weird. Yeah, we don't want to mess with those. Because something is wrong. And if something is wrong, other things may be wrong. Yeah. And so then we're just going to click now to build out the front end, to build out the beginning of our sequence. We're going to click a bunch of reverse reads. This is like our TED Talk moment where you say, oh. We're going to find reverse reads that line up with the front of the thing we searched for. Oh, sorry, reverse reads. There we go. Because we know that these are paired end reads. Go back and look at the picture of the fish if that's confusing. Yeah, or like Google it. AI is pretty good at these things these days. If we find one with a reverse end here, we know there exists a forward end somewhere near or far to this. And so every time we click a reverse, we get a chance at a different forward. And if we get several of these things, we can build several steps at once. This is only how you go fast. If you cannot get your head around that, click a forward read, move 7 bases at a time, and by the end of 300 searches, you will be done. Yeah, this is only an efficiency thing. So I'm finding reverse reads. Let's see. Reverse read is going to have a big number here and a little number here, and it's going to match up to the beginning. So there's the beginning, and what we want to look for are different numbers. The way the databases are made, if they have the same number, it may actually be just another copy of the same fragment. Sometimes people make copies before they do the sequencing. It's annoying. So to make sure our chances are as good as possible here, I'll click a 145, I'll click a 150, 149, that's great. 143 is super. 139, cool. And whatever else. So 130, why not? Round it out. All right. Now I can go back and look and see what each of those was. So my first 145 here was a 0.2. And I can say, okay, 0.2 is my reverse read. No. Yeah. There we are. Sorry. Yeah. What we want are the forward reads where we know where the reverse is, and the forward is going to be somewhere out to the left of it. If you want to be smart and go fast, who doesn't, right? What you can do is take a sequence that's in here that they'll all have, mentally make the reverse complement. So you're going to go backwards, and every time it says C, you're going to write G. And every time it says A, you're going to write T. Yeah, got it? Okay. So this is going to be GGCG, TCTCG, GGCG, TCT. And then we'll do a search, GGCG, TCT. There we go. And so that's going to be the reverse one. That's the one that we don't want. The forward one is going to be the other one. So we're going to select the forward one, and we're going to go, and we're going to paste those all into our nice little document. We're going to stack them all up, and so here's a bunch stacked up. Once you have them stacked up like that, we're going to take our seed, and I usually stack these kind of in front of the seed, sort of between. I would make a little space here. I would paste all these right in there because that's a good spot. We're going to look for AT, ATA, GCT, GCT, because that's the first couple of sequences right there. And then we're going to see, does that also occur? Oh, look at that. It does occur. Now for me, I like an easy life. So I'm going to pick the one where we have that match and we have the most sequence before it. So it's going to be this one. And so all of this sequence is going to be additional. And we can add that to what we got here. Oops. There we go. And so boom, we've just extended it. And then we can see if we got lucky and we can extend it again. So CTCCGC. Okay. There we go. And look at that. We can get this sequence and we can get all this extra stuff. All right. And then we can paste that right there. You do not have to keep the little colors. I'm just doing the colors so you can tell what's new and what's not. So here, I'm going to make that go away because I don't want to mess up the instructions. But that's how you do it. You find the matching bit and then you join them up. Boom. Yeah. So you don't want any sequence to be doubled. You don't want the sequence to be repeated twice where they overlap. You just want it to be a nice clean thing. Yeah, I don't know. Yeah. Try it. And you can compare your results to the one that I did. And if yours don't look like the one I did, then you messed up, I think. Yeah. Unless I messed up. But I probably didn't mess up this time. Sometimes it happens. I don't know. It's no big deal. You just go back and do that part again. Okay. Once you've built forward as far as you can go and just keep searching until you run out of sequences that will extend it, then you're going to go to the other end. So we'll go down to the other end. And we're going to do the opposite of what we just did. So here's the other end. And so now we're going to take, again, it's going to be the ones with the big number on the end. And we're going to get different big numbers. So we got a 150, we got a 129, we got a 127, we got a 126. Oh, sorry, that's the 6. There we go, 126. And each of these things is going to open up. I'm doing, the little clever trick is if you do Command when you click if you have a Mac, or Control when you click if you have a PC or Chromebook, it will open in a new tab. If you just click the link, it actually messes with the formatting on this one, and then you have to spend about a minute reloading the page to get the next one to open. Yeah, I find that annoying. Your results may differ, yeah, but I think you would find that annoying. So open a new tab, or like right click, open link in new tab if you're like bad at keyboard stuff. Yeah, because that's still faster than reloading the page by a lot. Now for this one, we want to find reverse reads. So we picked forward reads because forward reads have a big number on this side. And we want the reverse read that goes with that. It's part of the same pair because we know we have this one and it's right at the end. So we know that the reverse read is somewhere over here. And we'll extend our sequence in the way we're trying to extend it. We're trying to make the front grow more toward the front. We're trying to make the back grow more toward the back. Rewatch or try the example, see if you can match mine. Yeah, it takes a minute to get your head around that, and that's okay. But you need to get your head around that, otherwise it's just so slow. All right, so now let's just pick this sequence, and we can say just search for that. Find paste. Okay, that's not one of the ones I just opened. What? Okay, I did something wrong. Oh yeah, okay, it's one of these, sorry. Oh, there we go. I don't know why. All right. So if that's the forward read that we just searched for, then this is going to be a reverse read. That's just a quick way. You can always remember the number, like it was 6729.2 was the forward, and so 6729.1 will be the reverse. That is always an alternative that is available to you, my friend. Yeah, so double click it, copy it, paste them all in there. Here's a bunch of them. Now all these things are going to be reverse reads, so they all need to be reverse complemented. That's okay. That's easy. That is an easy fix. You're going to go to reverse complement dot whatever, this site. It's linked in the instructions there. You either got to paste them all in one at a time, or you can paste them all in and you can, here, let's do it. And this is only a time-saving tip. It'll work if you do it one at a time, if one at a time is your speed. And you're doing this for the first time, don't feel bad about, going in slow mode for the very first one. So you can replace that. This line is just the title, and replace it with a lowercase n, because those will stay intact. Lowercase n, because reverse complement tool can only do one at a time. It's annoying, I know. There we go. Okay, and now you hit submit. There we go. Copy, and you take that over here, and you're going to paste it like so. Here, like so. Paste. And then everywhere there's a lowercase n, you just delete that and hit return, and they'll be all lined up because they're all 150 apart. And there we go. And so then that's what I've done, and so now we can undo that because I don't need this. There we go. These are the possible extensions, and so now you're going to search with the end of your sequence. ATGCCAATT, for example. And let's see if we can find one that we can use to extend our little reach. Look at that. And so now I'm going to pick the one that gives me the most on this side, because this is the side I'm trying to build. So that's a good one. And we just add that onto the end. And then we see if we can add onto the end of that, etc., until we run out of sequence that we've copied. So if you get like 6, 8, 10 of these possible sequences with different numbers, you can sometimes build out 300-400 on either end. The whole virus is only 15,000 to 50,000 long. So if you can build out 500 each round, then to make a 15,000 nucleotide virus, that is 30 searches, and you're all the way at the end. If you can build out more than 500, which is doable, it's actually going to come down from that. It's going to be fewer searches. Probably won't be more than 100 searches, but whatever. It depends on the pace that you can go at. Once you got that, then you're going to do the next round. So you're just going to repeat step 4 over and over and over again. I'm going to grab the first 2 lines of this, and I'm going to grab the last 2 lines of this. I like to put it in this Courier New font because it's monospace. Everything takes the same amount of room. So you can tell you've got exactly the right, number of these things. In between them, I'm going to put a couple lowercase n's. That's my just little all-purpose trick that always works. And then you put this in, and you go back to your blast search. You're going to search in the same database, same SRA. So the only thing you change is this. You paste that, and then you hit go. And everything, it'll keep all your settings. And yeah. Now you got a new one. You can close the previous one. You can close each of these when you are done with them. And that is the entire procedure, start to finish. You just keep going until you run out of sequence. If you get a complete virus, you will have a poly A tail at the end, or a 3' untranslated region. You have to run it through a translator. You have to run it through some bioinformatics software that predicts what kind of protein you're dealing with. I will do all that. Don't worry. You're the little robot that's building the sequences out. That is your function here, and that's what will get you loads of points. And as many as your brain can stand. I'll do a couple of these on a typical weekend morning because because I like it. It's like Sudoku. It's like, it's like doing a jigsaw puzzle. It's the same human instinct. You're taking something broken and you're trying to fix it. You're trying to extend it. You're mapping a new part of reality. Like when you go into a game and you're in Zelda or whatever, and you're mapping a new little sub kingdom or section or something. Yeah, that's exactly what we're doing. Only it's a virus that's making something sick. In this case, an axolotl. So, final thing. When you're done, you're going to do one more round. So you're going to end up searching for this one, see if you can extend the thing that we already made. If you can figure out how to do that and do it successfully, send me the thing that you just built so that I can see that, like, yeah, okay, this person has made it longer and it does kind of match with what the virus is supposed to be. There's plenty of little tips and tricks in there for like the ways that it commonly goes wrong. Read them. They're in the correct section where right after you do the thing, it'll say, hey, and if it goes wrong in this way, this is how you fix it. Yeah. Once you have sent me a like one-step extended version of the ambystoma coronavirus, then I will send you a new organism and a new database and a new seed RDRP, and you will repeat this process. You'll go through the first 3 steps one time only for your data set. And then you will just repeat step 4 over and over and over again. Just bam, cycle through it until you run out of sequence, till you run out of road. And then you're at the end. You did it. You built yourself a virus. And you've contributed something to science. And you got some bonus points. Like, that matters? Yeah, that matters less than all the other things I've said, but maybe it actually matters a little more to you right now. And I get it, so that's fine. You're helping me, I'm trying to help you. Yeah, we're helping each other. So there you go. That's it. I'm here, and I'm here to answer questions. A lot of questions you'll be able to Google, because you'll be able to get to AI Google thing faster than you can get a hold of me. Yeah, but I am here to help. And a lot of it, if it's not working, you pasted something wrong, or you did something wrong with the blast settings. Like, I'll sometimes forget to change that to SRA and paste in the thing, and whatever. It's dumb mistakes. I have made those dumb mistakes. Don't don't think I haven't. I make them all the time still, and it's okay. It's just a little blip, and yeah. 5 seconds, you notice what you did, and you're like, okay, fine, whatever, and you fix it and you move on. There you go. So let's build a virus, not like really in a way that hurts people, but let's reassemble a virus that actually exists and is probably ruining something out there's day right now, and is related to viruses that are actually super dangerous to human health, that we probably ought to know a little bit more about. Yeah, and it's a brand new virus. It's potentially publishable, but there is so much left that has to be done to make these into a publication that I don't think you have time for that right now. But it's a independent research project that I have undergrads do in my lab all the time, and yeah, that would be the pathway to do that if you want, and I'm here for that. So thanks very much.